T'as un des deux. T'as un livre dit check couvrez-vous.
Allô? Allô? Mais non? Oui, il est coupé celui-ci. Attends. Ah non, non, non.
À certains endroits. C'était Ruud, la première chose qu'il a fait dans son bureau. C'est son blanc.
Allez, bonne séance. Ladies and gentlemen, good morning. Today we're going to do this session in English. The 2022 has passed quite quickly, at least for some of us, so we are already in December, amazingly. It's like yesterday that we were in January. And today we're going to have 3 speakers. Before introducing them properly and their fields of expertise, I will ask Madame Vanessa Costanzo to explain us how the digital world will function in parallel to ours.
Professeur, docteur, mesdames, messieurs, bonjour, good morning professors, ladies and gentlemen. Comme toujours, 10 minutes seront consacrées aux échanges avec l'orateur à la fin de chaque conférence. Les membres de la compagnie peuvent poser leurs questions via le chat Zoom. Ceux qui le souhaitent pourront poser leurs questions en live. Voici ce que vous verrez alors apparaître sur votre écran, si ça veut bien se faire voir. Alors, si vous nous suivez sur YouTube, vous pouvez nous laisser des questions écrites que nous transmettrons aux orateurs. N'oubliez pas de préciser votre nom, votre prénom et votre institution. And I will now briefly summarize this in English. If you are watching us on Zoom, you can either write your question in the Q&A section or agree to ask it live. YouTube viewers, unfortunately, can only ask their question through the chat. Clonal tissue heterogeneity increases with age. It can alter tissue function and predispose to some diseases. Our first speaker focused his research on driver mutations, leading to an increased risk of blood cancer and cardiovascular mortality. Mr. President, you will introduce him.
So it's a really great pleasure to introduce Professor Paresh Vyas, who's a professor of hematology and a consultant hematologist at the University of Oxford. He is a group leader at the MRC Molecular Hematology Unit, and I think the scientific director or medical director of that unit, and the Department of Hematology of the Wetherell Institute for Molecular Medicine at the University of Oxford. He has been educated at the Cambridge University in Christ's College, then medical school at Oxford, and then postgraduate qualifications in both medicine and biomedical sciences. He is a leading expert in the molecular basis of acute myeloid leukemia and in general in hematological diseases. And without wasting more time, we're looking forward to hearing his talk. Well, while we prepare that, it's a pleasure to actually read the title of the talk, Clonal Cell Heterogeneity in Tissues with Aging Alters Function, using whole genome sequencing and single-cell sequencing to study these processes in blood cells as an exemplar.
May I interrupt you for a second? Because seems to be a problem online. People are just notifying the fact that they do not see anything. They hear us, but they do not see us. So we are going to try to find a solution. Donc, en français, on est en train d'essayer de chercher une solution. Nous n'étions pas conscients du problème. Merci de le signaler.
Oui, oui, normal. Oui. Oui, oui. Oui.
Mr. President, since people can hear us and listen to us, maybe just a question for you, because you have been the president for a year. This is coming to an end.
Yes.
So how was this experience for you?
Well,
Let's
thank you.
start
That's
with
a
this,
very
huh?
good question. Thank you for the question. Excellent. Well, I have to say that it was a privilege to work with our colleagues and to learn a lot and meet people that I would have never met otherwise. So it's really exposure to different fields of medicine, forcing you to get out of your narrow area of interest. Of course, certain things took much more time, and because I didn't have the time, sometimes I didn't do them very well, because if you don't prepare, always you get in trouble. But in the end, I think I learned a little bit. And I think it was a great privilege, and I learned a lot. And I can translate this experience at the Federation of European Academies of Medicine, because now I have the experience of how it is to receive all these inquiries from all these people and to have to respond. Because before it was easy, you just ask everybody things. Now I know it's pretty difficult. You have to really trim down the questions to really the key questions. So I think it was a great year. I think we have met a number of people, especially also new members, because every year comes with new elections. So you have to study a lot of interesting CVs, interesting discoveries. So you, again, learn a lot of interesting things. So I hope that, and also had been in touch with the Flemish Academy, with the Academy of the Theresian Academy. So it's, you learn a lot of things. So it's an educational process.
How did you choose the people you invited today, the speakers?
Well, it's...
After that, the third question will be a song. I'm sorry. That's the second one. I have no question left. So the third will be a song.
Well, it's a very selfish thing. It's the most, to me, at the moment, I'm interested in... So I think that there are two fundamental questions in biology that make people now have difficulty getting progress. One is the protein folding code. We don't know how proteins fold, why they fold, what are the rules, and that is why we can't make drugs. All that we do is screening empirically. We cannot predict the proteins in the chain of signaling, how they interact. And so this is one of the big questions, and Steve Smith is an expert in that. And then the other question is the chromatin folding code. How does the DNA plus protein in the nucleus assume the spatial form that allows differentiation? That is the choice of enhancer-promoter interactions that gives you specific gene expression. And the other two are experts in that. One of them is really very fundamental, Ruud Delwel, who discovered some fundamental translocations that induce leukemia and how these enhancers work. Exactly the key question that I'd like to really learn from him and get some ideas, so it's very selfish. And Paresh Vyas also has a broad view on acute myeloid leukemia, the disease we're interested in, to learn how to prevent in our chronic diseases and the progression. So it's very selfish.
So I guess you are lucky. Because online, apparently, and I would like people to confirm that, they see the image. So we just have a problem in the room. So either we take a break in the beginning of the session and we try to handle this. Alexandre.
Now, this is a novelty. We never had that. We had other technical issues
that were linked
to the fact that people couldn't hear anything, or it was an echo that would completely blur. But we never had the problem of not being able to transmit the slide. That is a new thing. And we are here to do research, so always new things are good.
You have to test everything under your president's field.
Yeah. Right.
Désolé pour ceux qui sont en ligne, on est en train d'essayer de régler le problème. Ça vous laisse le temps de prendre un petit café à la maison ou de nous rejoindre si vous voulez. Ah oui, mais avec le métro, ça peut être rapide. Hein.
Well, at MIT, I have seen once when Professor Robert Weinberg had this problem at MIT, and he was forced to give 1 hour talk without any slide. But I'm not going to do that to our guest. But it's not happening only in Brussels. It has happened before. It's called Chalk Talk.
Ça, on n'a quand même jamais vu. Hein. Est-ce qu'il prend une photo?
Qu'est-ce qu'on fait? Parce que c'est vraiment incroyable. Ça, j'ai jamais vu ça.
Georges,
Yeah.
peut-être un petit verre de vin pour détendre Georges. Peut-être un peu.
No, but imagine that the projector has a problem. We are screwed. There is no projector, reserve projector.
L'autre qui peut être.
Sorry.
Voilà, on nous demande 5 minutes d'attente pour pouvoir régler le problème. Donc en ligne, ne partez pas. Restez avec nous, s'il vous plaît.
Because we cannot.
Oui, j'ai tout suivi. J'ai tout suivi, mais on n'a même pas de caméra, rien. C'est la sortie du radio vers le projecteur. Parce qu'on n'a même pas de caméra, on peut avoir une caméra.
So it's here. Here it is. See? It works here. It just doesn't work.
It's not communicating. It's not communicating. Is there a pointer? Look, in the end, will there be a pointer? Because I do need a pointer. So what are you doing over Christmas, Ruud? Family?
Yeah, my family from France comes over. My son lives there. So he
They're
is
meeting at your place?
Yeah, but because they haven't been over in 3 years, right? Because of COVID. So that's so we will be for the first time everybody together, all the children, grandchildren. Yeah, we look forward to it.
Are you doing all the cooking?
Huh?
Are you doing the cooking?
No, I just go to the restaurant when they're all there. No, but we do cook, you know. And then we always have New Year, New Year's Eve. So there's this very common thing in the Netherlands. We make those called oliebollen. It's what do you call it? It's a bowl, you know. It's you make it in fat. It's with raisins and I cook many of them, and then the whole neighborhood I invite them to come over, and then you make a glass of wine during that's during the day, 31st, and then the whole family comes, friends. All oil balls we call them, oil balls. So you make them your own.
So it's fat and fruit?
It's pretty fat. Yeah.
It's fat and fruit?
No, yeah, fat and fruit.
And then you go back when? When do you go back to work? When do you go back to work?
No, today. No, tomorrow. No, tomorrow, Monday. I mean, no. Yeah.
No, after Christmas.
After Christmas. Yeah, during Christmas, you know, I like to go to the lab, you know, because there's nobody there. I just do some stuff. Although, you know, since COVID, I really have an office at home, so you can do it. I like to be there, you know, just do some writing. Really have to finish papers. And during COVID, we were very efficiently writing papers, submitting all of them. And now, you know, we are in the lab again, and, you know, start traveling, get invitations, and I realized, you know, how good it is to not travel, and you could not do so. Because we always say, you know, to people, you know, okay, you know, give it, you know, I read it, you know, in the plane, I do, you know, my hotel. It's not true. You don't. I mean, when I'm in my hotel, I, you know, meet friends. You go downstairs, you go to the bar, and, you know, you do talk. You don't do it, you're not efficient at all. It's efficient.
It is inefficient. It's very inefficient.
But, you know, it's also so nice to do, huh?
Well, it's nice to
Travel
meet
and
people.
meet people.
It's nice. We're social people. You had a fantastic lecture series in Erasmus.
Yeah.
Really good.
Yeah.
Got
We still go on with it.
You organize it, right?
That's right. I have to send a tweet because so Monday we have the last one with Paul Yu. Yeah, that has been successful. Really. You know, we had up to we did it weekly, right, during the pandemic, and we had 300-400 attendees every month. Yeah. And then there were the people, there were quite a number of people that did it, in the library, they filled up the library, and there were this one collection where 10, 15 people watching. So,
Yeah. Well, it gave firstly.
Well, Cambridge did the same thing. That was also with the epigenetics.
Yes.
There was once a month.
Once a month. Brian was involved with that.
Yeah, I gave a talk.
He had a good story at Ash on the condensate, but I didn't quite agree with the science, but the fellow who gave the talk, the lady who gave the talk, gave a good talk.
I don't know about those condensates. I'm really afraid that we're also looking at I did a lot of EVR1 overexpression in the past, and I always found, those nuclear speckles we call them. But they were the result of overexpression. If you just look the patients out, and you do Stony, you don't see that. And there was one talk about NPM1, mutant NPM1, I don't know if you've seen that. Also about speckles. They really showed the more mutant NPM1 they gave, the more
Yes, I see.
condensates they got. I thought, and people were really excited about it. So if this is really a real thing, why didn't Bruno Fellini, when he for the first time really showed, right, because he identified mutant NPM1 just by pathology, he really stayed here, and he found that it went to the cytoplasm.
Exactly.
He didn't see anything.
Exactly.
That was the story.
Exactly.
And now they have all those overexpression systems, and you see that. So I think we're looking at an artifact of the system. And I think there's nothing.
But we don't even know what it does in the cytoplasm.
No. Also, well, there is beautiful the work now, and that is not overexpression from Scott Armstrong and about the two new papers
In cancer discovery.
Cancer discovery about DNA binding, right, which is just in the cells that natural mutant expression. Then you see most of the stuff in the cytoplasm, but there's still some left that binds DNA. I don't want to know how that works, but at least this is not. But the condensates, I don't like it. I'm not working on it. I'm not going to do it. I think it's.
So where are you taking the work that you showed with Ash now? The work you're going to present today? Where is that going?
You mean the EVR1 C2BP interaction? So we still have to finish a few things, but then you mean where we're going to send it to?
No.
Where are you going after?
Yeah,
So we have a couple of assays developed now. And one of them could be just a simple alpha assay, that is a biochemical assay, where you have purified protein and you can measure with fluorescence the interaction and loss of interaction.
So it's like a Fred assay.
Sort of Fred assay. But this is really biochemical. And we have
This is
another
with
assay.
purified protein.
Purified protein. Another one is where we really have some sort of, you may call it FET, in the cell, protein-protein interaction assay. It's called a mapping. And where you can really apply to screen, compounds that interfere between EVR1 and C2. Another possibility is just there are chemical libraries, with millions of compounds that are now part of it. Okay, so every chemical has its own. and then fish in the library and isolate the compounds
That bind.
and then sequence.
But how much, okay,
Because what you then could do is not only see whether you can interact, interfere with the interaction, you can also then, if you have a compound that binds specifically to the then you can make a protein with that
yeah, of course,
and degrade it.
yes, yes.
I think that would be a cool thing to do.
So the screening assay, so you have many molecules of EVI1, you have many molecules of each of your libraries, and then the sequencing, the DNA barcode will be short, right? So how do you sequence it?
That's a good question.
Right? It will be a 10
It should be
repeater.
No, it will be larger, I think. And I think what will be happening, so it will have, they will all have the same.
Do you need to be here?
No, it's okay. Isto da che ce câble ne va pas.
It's a cable that's rubbish.
Huh?
It's that cable, I think. It's the HDMI cable. That's the problem. Yeah.
I think the problem now is, now we are here, but now we are not online. He speaks English, he speaks French,
Romanian.
Romanian.
But you know, he grew up in Paris.
Huh?
He grew up in Paris.
Yeah. But it's because he lived, he worked together with Weizsäcker. Yeah. He was on that famous.
Educated. James et al. V617F paper, yeah?
Yeah, the one that we had so much discussion about. Not their paper, but the other papers. That was nasty.
Tony's retired.
Huh?
Tony's retired.
Did he really?
Yeah. So Bertie took over the Stem Cell Institute.
Okay.
Bertie got it.
And
Brian,
the department.
The hematology.
Yeah. Oh, really?
Yeah. Brian will be good.
Yeah. He's doing a lot, by the way, because he's also quite active in EHA. So he came in and I immediately stepped out. He said, if I would have known that you would have stepped out, I would have done it. Because he's really on his own now.
What's his position in EHA?
He's in the board. Not an executive board, he's in the board.
Voilà.
Looks like an image.
So do you have a lot of administration to do?
I did a lot for EHA, and then, first at some point, I did EHA, Dutch Cancer Foundation, but there was a couple of things that we had to do a lot for. But, I'm out of all, and I'm not going back. Just only in the last couple of years, I just wanted to decide. So I was at St. Jude's, and so I told them that I was going to retire.
Yes.
exemple, là, ils ont une image ici. C'est ce que le boss veut. Et je ne sais rien faire.
Non, mais donc... ici, on doit repasser... Si.
Speaker, Professor Paresh Vyas, to go back to the presentation. I think we will start. We apologize for the technical difficulties, which are recurrent and increasing in intensity with session. But of course, this is the reality, so we have to live with it. Right. Okay, good.
Well, it's a huge pleasure to be here, and I'd like to thank the Academy, and I'd like to thank Stefan in particular. Great. Thanks. So, as I said, thank you once again, and I really hope this is going to be worth the wait. So, I'm a hematologist by training, and today I'm going to tell you something around whole genome sequencing and two processes that occur in every tissue in the human body and in all organisms that lead to the expansion and contraction of clones of cells. And the two processes are neutral drift and clonal selection. And I'm going to be talking a little bit about stem cell dynamics. So many tissues are rejuvenated, replenished by stem cells that differentiate into more mature cells. And when each cell differentiates and produces its progeny, those progeny can expand and contract to produce a clone of cells. And so it's these two processes of neutral drift and clonal selection. I'm going to use blood as an example, but what I'm going to be talking about is probably going to be common across many, many, many tissues. So when you start off with the first fertilized egg in embryogenesis and the cells expand to form an organism, each time a cell divides, there are genetic mutations. And there are probably 3 or 4 genetic mutations in every cell division that occurs. So even when you're born, you are a mosaic. Each cell is different from another in your body. So you are a genetic mosaic. You're not a homogeneous genetic organism. And so these somatic mutations really are seen, and I've shown them here in gray. So every time a cell is dividing, here you get 1 to 2 mutations. And these mutations, if they occur in a long-lived cell, a stem cell, then the mutations will be propagated to all the progeny of that cell and will be retained for a very long time. So you have genetic cell heterogeneity. Another term for this is somatic mosaicism. These two words are interchangeable. And somatic mosaicism can have functional consequences. So in a tissue, you have all these different, genetically different cells. Now, if these mutations occur in genes that change the function of a cell, then that tissue is going to have an alteration in function. And so you can alter function through somatic mosaicism. And if the mutations are driver mutations, i.e. they lead to expansion of that clone of cells, this is the first step in the multi-step process of carcinogenesis in most organisms. The other thing to note is somatic mosaicism increases with age. So as you age, you have more and more cell divisions, you get more and more genetic changes. So your genome is really very different today to the genome you were born with in all your different cells. So more mutations are required with time. And there's increasing numbers of people in the world working on aging, and they're trying to understand how somatic mosaicism alters tissue function and leads to the phenotypic properties of aging. So clones of cells with genetically distinct features can either expand or contract, as I mentioned. And if they do that stochastically, by chance, these mutations really are affording no selective advantage. We call this process neutral drift. Clones can also expand if a somatic mutation affords a selective advantage. So this is Darwinian, this is what Darwin taught us, and we call these driver mutations. And here there's a clone that's expanded quite early in embryogenesis, creating a large clone of cells. And if that mutation in red affords a selective advantage, we call that clonal selection. So in blood, this was first demonstrated in 2014 in the New England Journal of Medicine by the group of Ben Ebert and Steve McCarroll, working both in Boston, both groups working in Boston. And then there was a group at WashU in St. Louis at the same time who published. They showed that in most people who have no phenotype, they're just ordinary citizens walking in the street, they could have mutations in their blood that lead to an expansion of those cells. And it's a driver mutation that's providing a selective advantage to that stem cell. And so the progeny of the stem cell come to dominate the blood. This resulted in a 10-fold increase in blood cancer it also resulted in a twofold increase in cardiovascular mortality, both myocardial infarction and stroke. And the mutations that were identified that were common were in genes that I've listed here. The names really don't matter, but what is going to be relevant for the talk that's to come is that the two most common genes that were driver mutations here really affect DNA methylation. This affects chromatin structure. And this is a signaling molecule, and these two molecules here affect RNA splicing. So you can see fundamental processes in the cell were affected by these mutations, these driver mutations. The mutations were acquired in blood stem cells. They gave a clonal advantage. They're really myeloid cancer driver mutations, but also the same mutations alter the function of mature neutrophils and monocytes. And the field is now coalescing around the sense that it's the derangement of mature myeloid cell function that leads to the excess cardiovascular mortality. So you have a gene affecting both the very beginning of blood cell differentiation and the very end of blood cell differentiation. Since the findings in blood, a large number of studies have shown that somatic mosaicism probably occurs in most organs. It's been demonstrated in the skin, in the skin, the liver, the endometrium, the prostate, and the large bowel. And in all of these organs, these driver mutations are pro-cancer and also they're pro-inflammatory. So there's a common theme emerging. So I now want to go back to blood. Clonal hematopoiesis is the name given to this process where a driver mutation selects a large clone. And the open questions in the field is, does expansion occur by selection, neutral drift, or both? And how can one distinguish these processes? When are these mutations actually acquired in life? So the original papers studied individuals in their 50s, 60s, and 70s. But when was that clone really born? When did it first arise in life? And once the clone occurs, does clonal advantage arise from increased number of stem cells produced by the clone or by other mechanisms? Really, we need to find methods to assess the trajectory of clonal growth in individual human beings. That was the series of questions we set ourselves. So I should just put this slide in really as a conceptual slide. So somatic mutations really act as a clock. So when you acquire that mutation in time, so this is increasing age, it really is a timestamp. It measures that point in time. And then if that time is early in development, many, many cells will have that red mutation, as you can see. You can see this blue mutation is acquired later in time, and there are fewer cells with the blue mutation that I've shown here. And here, if you draw the total number of mutations acquired by all the cells, so here you can see a total of 125 different mutations acquired by this cell. The red dot is present in more cells, and so we say the variant allele frequency, i.e. the frequency with which the variation occurs, is higher. The total number of mutations associated with the red cell will be smaller because it's occurred earlier in time, but the frequency with which cells have that mutation is higher. So the higher variant allele frequency, lower total number of mutations. This cell here will have accumulated many more mutations by the time you have the blue driver mutation, but there are fewer cells with it. So the variant allele frequency is lower, but there are more mutations associated with the blue clone here. So you can plot variant allele frequency versus total number of mutations in a graph like this, and the higher the variant allele frequency, the earlier in time that clone was born. So just by measuring the variant allele frequency, you can really put a place in time when that clone was born. So that's what I'm telling you here. And the co-occurrence of passenger mutations with the driver mutations really can be used to determine the age of the clone. So another way of showing this is here. So this is in development. You can see the mutation A and B, high allele frequency, total number of mutations associated is small. As you go later in life, now you're keeping the number of cells the same because you've passed through development. Now you're in adult life, you have homeostasis. If you require a mutation C and E, they have lower variant allele frequency, but more mutations associated with it. And then here you can see the clone associated with E dies out, but the other clones remain. And so you're now left with AB, which has occurred really at the similar sort of time, C, and then D and F. And so you get curves like this. You can change the curves by, instead of plotting VAF, or variant allele frequency, you can essentially plot the inverse, 1 over VAF, against the cumulative number of somatic mutations, and you get curves like this. So early in life, you have very few mutations, 10 I've drawn here, and then later in life, you have many, many more mutations. And as you get older and older, the curve bends upwards. And this is neutral drift. There's no selection. This is just the variation of clones by size over time, no selection. And indeed, we did whole genome sequencing on 10 normal individuals, and that's exactly what we see. So this is now modeled data. This is real data from real human beings. Here I'm showing you data from 10, but we've done now 50 human beings, and really the curves really look like this. So this is somebody in their 30s, this is now somebody in their 80s, and you can see the 1 over VAF curve versus the cumulative number of somatic mutations. You bend upwards. It's exactly like that. So this was in gray, the model, and here are the dots that are the real data. So you can then do some mathematics, and we collaborated with a professor of mathematics at the DKFZ in Heidelberg, Thomas Hofer, and you can really ask very simple, you can use, as I've shown here, some very simple assumptions. You can have a division rate of lambda that leads to productive cell division. You can have a mutation, and you can really determine the rate at which this mutation is fixed. Or the cell can die, and you can have a death rate, a delta. And early in development, you have more cells growing than dying, so gamma is greater than delta, and the total number of cells is less than the total number of steady-state cells that you end up with as an adult. And then in adult life, death and differentiation are balanced, and you have a total number of steady-state cells. You can then use the data that we had in the previous slide that I've showed you here to work out how many divisions are occurring per every stem cell division. And this is data from human beings of different ages, and you can see that every cell division you get 1 to 2 mutations every time a blood stem cell divides. You can then work out the number of divisions per year. There are 1 to 2 stem cell divisions per year in human beings. These are blood stem cells. And then finally, you can work out the number of active stem cells. And they're between 1,000 and 20,000 active stem cells in most human beings, regardless of age. Now, what happens when you get a selective mutation? I've shown you this clonal hematopoiesis in a previous slide, where a mutation really expands, actively expands the clone. What happens there? Well, you can model that too. So here you have a clone which has a selective mutation, D, and what happens is all the mutations that were present in that cell D, they also expand. So here is D, and as that cell continues to expand and dominate, all the mutations associated with D increase in number. So you get this step change in the curve. And this is exactly what we see in human beings who have clonal hematopoiesis. The total number of somatic mutations increases. So this is the cumulative sum of somatic mutations. These are patients who have driver mutations that give rise to clonal hematopoiesis. You increase the number. So the model and the data fit. And then over time, as you age, you then go back to your normal curve. So this is the VAF curve, this is 1 over VAF. You'll get something like this. And that's exactly what we see. This is a patient who had clonal hematopoiesis. This is the cumulative number of somatic mutations, 1 over VAF. This was the model, the mathematical model that predicted what we would see. And this, sorry, I'll go back. This is the actual data. The data points are here. So our mathematical model really does predict what selection looks like. And this shows you the posterior density distribution, i.e. the likelihood you have a clone, and it's pretty accurate. You can then measure the clone size either by the population genetics model that I've shown you here, or by directly just measuring the number of cells that have the mutation, and the model and the actual clone sizes measured by measuring the mutation really agree beautifully well. So now we have a model of drift and we have a model of selection. And you can then infer when was that clonal hematopoiesis clone born and what are the number of stem cells. And you can see the number of somatic mutations in a selected clone is really the same as normal. You can see that the stem cells divide 3 times as fast when you have a driver mutation. You can show that the number of total active stem cells is 3 times greater when you have this clonal driver mutation. This is the average, and this is individual data points. And then finally, you can age the chip clone. And in most people, that initial driver mutation normally occurs between the ages of 20 and 30. So really, most of us already in this room will have a driver mutation in our blood cells that is expanding that clone. So this is the first genetic step in aging. Really begins in your 20s and 30s. And in some individuals, it can even begin in the first decade of life, which is really rather extraordinary. So in this part of the talk, I've shown you a new method for differentiating between neutral drift and selection. I've shown you that the number of stem cell mutations acquired per cell division is the same, whether you undergo neutral drift or selection. I've shown you that these selected clones in clonal hematopoiesis in individuals in their 50s to 80s really start 20 to 40 years earlier. And in occasional cases, they really acquire very early in life. The division rates and stem cell numbers increase when you have a selective driver, and the clonal expansion occurs in part through increased stem cell numbers. I think this will be true in most organs. So in the liver, in the kidney, in the skin, in the uterus, all of these processes will play out. And I think we now have a very simple method, a whole genome sequencing method on bulk cells that gives individual level information. So it gives information for that individual human being. And really, it's a potential to map trajectories of clonal growth within individual human beings. I just want to change gears, and I don't know if we've got time. We were late starting, I don't know, we have time. So in the second part of the talk, we're going to use that same biological process of clonal expansion, clonal hematopoiesis. Now, instead of looking at bulk populations of cells and looking at the growth of cells as a population, we're now going to drill down onto individual single cells. And the cohort of human beings we've studied are aged match controls, individuals who have a mutation in this gene DNMT3A that gave rise to selective clonal expansion, selective clonal hematopoiesis, 4 individuals who had a mutation in a different gene called TET2 or TET2. Both of these genes affect DNA methylation and both give rise to clonal hematopoiesis. And then, interestingly, we had one individual who had both mutations, both a DNMT3A and TET2. And as I'll show you, they occurred in completely different cells. And so these two clones were competing with each other in the same human being. And we did single cell work here. So we take individual blood cells from the bone marrow, we separate them, it's called single cell sorting, into a plate. So individual wells would have a single cell. Then we genotype the cells, meaning we could identify whether they had the DNMT3A mutation, the TET2 mutation, both mutations, or were completely devoid of these selective mutations. That's called targeted genotyping. We analyzed the entire RNA transcriptome, i.e. all the RNA species. Using this method, we can identify 6,000 transcripts per cell. And I'm not going to show you today. We also looked at chromatin structure. Today, I'm just going to focus on the genotyping and the RNA sequencing. We studied 13,000 single cells, mainly stem cells, but we also looked at mature cells. There were 18 mutations in total across the 10 chip samples. Today, I'm just going to show you data on these patients here. It's a really high fidelity data set. So most of the cells we could genotype, most of the cells we could get really high quality RNA data and also data on chromatin, but I'm not going to talk about chromatin today. These are really novel technologies that are really changing our field. We're now getting, the world is getting a really unparalleled view of the DNA and the RNA within individual cells in human beings. And I think these techniques have already revolutionized biology and will continue to revolutionize biology. So what did we find? Sorry, it's not moving forward. I don't know why. Let's try here. No. Maybe the next slide is too big. Okay, great. Actually, I want to go to the next slide. That's what I need to see. Oh gosh, this is not turning out very well. I think this is a Mac PC issue. We're not seeing most of the data. I'm missing half the data. I'm really sorry. I think maybe I should just talk because the data's not coming through. But essentially, with the DNMT3A mutation, what happens is most of the impact of that mutation is to expand stem cells. It has very little effect on mature cells. Most of the clonal advantage occurs in stem and early progenitor cells. By contrast, the TET2 mutation, which is also clonal hematopoiesis mutation, has a major impact on myeloid progenitor cells and mature myeloid cells. And that's where most of the expansion occurs. Expansion does occur in the stem cells, but much, much more in the mature myeloid cells. And the RNA sequencing data shows you very clearly why this is. There is a deregulation of key transcription factors that really control the production of mature lineages, and in particular a transcription factor called CEBP delta, which is massively deregulated in TET2, but not in DNMT3A. The other thing you can begin to see is that the mature myeloid cells in TET2 in particular are really inflammatory, very, very inflammatory stem cells, mature myeloid cells. And probably that's the reason why you have this increased cardiovascular risk of myocardial infarction and stroke. The other thing I wanted to tell you was that there is competition between clones. So this is a human being who had two clones, a DNMT3A clone and a TET2 clone. On the y-axis is the proportion of cells. So this is a 77-year-old individual. 10% of his cells were TET2. 3% of his cells had the DNMT3A clone. Now remember, you have 20,000 stem cells. So to actually see a clone of this size, you really have to have a selective advantage. And you can see in the stem cell compartment, the blue is much bigger than the orange. There's a big stem cell advantage for the DNMT3A clone. In the myeloid cells, the orange is bigger than the blue. So the clone dominates later. So you see clonal competition within the same human being. And it differs in the different compartments during blood cell production. I just want to go to the next slide. The other thing we wanted to show you here is that if you compare the RNA signatures in the mutant cells, the mutant stem cells, versus the wild-type stem cells, you see hallmarks, the mutant DNMT3A versus control. You'll see the hallmarks of inflammatory response. But not only are the mutant stem cells abnormal, but the normal stem cells in the individual that has the mutation. So these two stem cells are together, mutant and wild-type in the same individual. Even the wild-type stem cells are different from control. So there is a non-cell autonomous effect on the wild-type cells as well as on the mutant cells. That's also true in the TET2 mutant case. So the entire bone marrow, the blood factory environment, is inflammatory. And one hypothesis is that the TET2 mutation and the DNMT3A mutation allow stem cells to clonally expand, to increase in an inflammatory environment. And I think the last thing I want to just show you is the conclusions from this talk. So, two mutations that afford a selective advantage, DNMT3A and TET2, they have different, distinct effects on differentiation dynamics. The selective advantage of one of these is primarily at the level of the stem cell. The other one really gives you a myeloid bias. And inflammatory pathways are upregulated in stem cells of people who have clonal hematopoiesis, and we've identified many of the gene expression pathways that lead to aberrant differentiation. I think this will be true in many, many organs. And indeed, we've seen this already in the liver, where exposure to high-fat diet, exposure to alcohol, leads to the selection of clones within the human liver that allow that liver cell to survive in a high-fat, high-alcohol environment. And these then clonally expand and ultimately lead to cirrhosis of the liver. So I think these processes will be similar in many organs throughout our body. Thank you.
Thank you very much, Paresh, for a fantastic talk. Under the difficult circumstances, it was even more important to deliver the message well. So I will start with a quick question. Can we flush them out? I mean, you showed us they can select Darwinian selection, but can we find ways to kill them?
So there is a large industry now, shall we say, of new companies, academics working with new companies to try and identify drug targets that will selectively kill, extinguish clones of cells that have this kind of selective advantage and that may have the properties of then developing into cancer and have properties to alter mature tissue function in a deleterious way. The big problem, and there is a big problem in the field, virtually everybody, all of us, will get these mutations, and not all of us will develop cancer. So there are multiple genetic and epigenetic and non-cell autonomous events that lead to the propagation towards cancer. And there are still an incomplete understanding of which clones of cells really are deleterious. So there's a lot more basic biology that we need to do, but there are already many, many companies working on trying to kill these clones of cells.
Pierre Coudy.
Thank you. Fantastically interesting. Somewhat connected to the previous question, is there any evidence of immunoselection?
Yeah, so I guess the field is still very immature to answer your question definitively, but the selection that we see in the TET and DNMT3A clones is not immunoselection in the way that there's an antigen that's providing a selective advantage to a cell, but we feel that the cells are able to survive in an inflammatory environment. So it's really inflammatory selection rather than immunoselection in the way of acquired immunity.
Maybe I have the microphone. Thank you for this nice lecture. I am a hematologist and mostly clinician. And when we analyze the marrow from all the patients, we very frequently find the mutation of the 2 and DNMT3A. And it's not easy for us as clinicians. When do you put the threshold between clonal hematopoiesis in older patients and the beginning of myelodysplasia? What is the threshold?
Okay, so for those in the audience who are not hematologists, let me just put a bit of color on that question. So as we age, often our blood production changes. We often become anemic. Anemia is very common in older people. You lose your lymphocyte responses to vaccination. You sometimes lose or reduce your platelet production, so you have a tendency to bleed and bruise. This is not uncommon as you age. And there is a distinct set of conditions called myelodysplasia, which are in some way overlapping with this reduction in blood cell production and blood cell function. But they're also characterized by blood cells, when you look down a microscope, that have very abnormal appearances. So imagine as a child grows to an adult, you know, we all wear a shirt or a tie, whatever it is, or one pair of trousers. Imagine wearing your underpants on the outside. Well, that's very abnormal appearance. That's the same with blood cells that grow abnormally. They look really abnormal. And we, through the history of medicine, have used the microscope primarily, and we define those diseases as myelodysplasia, the abnormal appearance of blood cells. So my colleague is asking, what is the relationship between the phenomena I've described here, clonal hematopoiesis, and myelodysplasia? Well, myelodysplasia is a morphological definition, as you know. The blood cells have to look abnormal. More than 10% of the blood cells in the marrow in each lineage have to look abnormal for that diagnostic criteria to be met. So if you don't have that morphological criteria, it's not MDS. That said, genetically, there is a clear overlap between clonal hematopoiesis and myelodysplasia. Many of the genetic mutations seen in clonal hematopoiesis are seen in myelodysplasia. But myelodysplasia also has other mutations, such as splicing factor mutations, which are not so common. It has mutations in transcription factors like RUNX1, which is a very common secondary myelodysplastic mutation. So there are additional second events, third events that you'll see, genetic events in myelodysplasia. Loss of chromosome 5q, loss of chromosome 7, so forth.
Yeah.
Thank
The question
you.
is...
We're going to alternate with the Zoom now, and we'll take one question from Zoom.
Yes, we have Professor Michel Goldman online. Professeur Goldman, votre micro est coupé et on ne vous voit pas non plus.
Est-ce que c'est mieux?
Oui, c'est mieux. Bonjour.
Voilà. So thank you so much for this very crystal clear presentation, which clarifies and emphasizes the importance of this complex phenomenon in aging and disease. So, as you said, we are born as genetic mosaics. So my question is, what do we know about the germline variants which might predispose to clonal hematopoiesis later in life?
Yeah, that's a really excellent question. So if I've just give it again a bit more color, what are we born with? What are germline variants, so present in every cell of the body, more or less, that can predispose to the acquisition of a genetic variant that gives a clonal advantage? And there are germline variants. And let me just step back. You know, when I first started medicine and you saw patients with leukemia in the clinic, they'd often ask me, you know, what are the implications for my children that I've got acute myeloid leukemia? And we would say, oh, it's a totally spontaneous, it's a rare thing. Don't worry, it means nothing for your children, etc., etc. But as we've learned more, particularly in the last decade, we begin to realize that there are families, in fact, not uncommon families, where you have a germline variant. And the most common is in a gene called DDX41. So we really don't understand this gene very well. DDX41 mutations are incredibly common, and they do increase the risk. These are germline, so they transmitted through families. They increase the risk of blood cancer. And so the dice are already loaded in those families to develop leukemia. There are other mutations which are much more penetrant, i.e. there's a much higher chance of getting leukemia, for example in RUNX1. So it's a much rarer mutation, but there are families who have RUNX1 mutations who will are more likely to develop leukemia. So in answer to your questions, we know some of the germline variants that predispose to blood cancer. I suspect there'll be germline variants that predispose to cancer in most tissues.
Thank you very much.
Thank you very much. I'm not a hematologist, I'm just thinking around. It was, well, beautiful and also unsettling what it might mean for genetic testing. So just realizing how biased we are. And it's very appealing to be talking about selective advantage. You mentioned survival, I suppose other aspects of reproductive advantage would be there. You emphasize cancer, aging, you also said survival, but then cirrhosis. My question is more general. I suppose that it might confer a general advantage to have this variation in terms of adaptiveness to change in environments. And my question specifically relates to development, where throughout our post-zygotic life, we will be exposed to changing environments, and how advantageous is it to have just this general variation?
Yeah, so if I've understood the question correctly, you're asking, so this is a pervasive process. Every cell in the body has the acquisition of mutations, and you're asking in post-zygotic life, how advantageous is it to have these selective processes occurring? Well, with every selective process, you really need to answer your question specifically, you really have to understand the biology of that mutation. So there will be some selective mutations. I've shown you DNMT3A and TET2, where the selective advantage is, you could argue theoretically, this is a hypothesis, that it is a beneficial mutation because it allows your blood stem cells to survive in a harsh inflammatory environment. And if you didn't get that mutation, maybe your blood cell production will be lower. So you could think of it as a benefit. But in the case of TET2, what I'm showing you is that not only does it affect survival and give you clonal expansion, it alters the functioning of the mature blood cells, the neutrophils circulating in your bloodstream. Is that beneficial? Well, it may be beneficial against a pathogen that you're more inflammatory, but it may not be beneficial for your cardiovascular system. So you really have to understand the biology of each mutation in the different cells where that mutation is functional.
Thank you for your fascinating talk. Do you have data about the sex difference in the process of differentiation.
Yeah, so is there a sex bias in the acquisition of these mutations? So not in the DNMT3A or TET2, but there are examples of sex bias for mutation acquisition, and we don't understand why these sex biases occur. So there are some blood cancer-causing mutations that have an extreme sex bias, often male sex bias, and we really don't understand the interaction of the absence of the second X chromosome, or indeed the acquisition of the Y chromosome, and all the developmental consequences that occur through that with selection. But there are powerful selective interactions with gender.
So now we understand why in medicine people say there are no diseases, they're just patients. Because every patient at age 60 will have 800 different mutations in their stem cells in addition to a potential driver, and those 800 may collaborate with the driver, and then the clinician has a lot of fun to distinguish between these people. So I'm moving now to the Zoom and ask our digital world.
Yes, the problem is that we lost Professor Van den Eyde, who was connected as a panelist. Professor Van den Eyde, could you please accept the request we are going to send you? Ah, okay, he's back. Professor Van den Eyde, your microphone is switched off. Professeur Van den Eyde, vous êtes sur mute, on ne peut pas vous donner la parole.
So while we're connecting, I just had a quick. So when you say that you have 14 mutations in each stem cells every year, it's not the same mutations, right? Every one of the 20,000 stem cells will have different 800 at age 60,
yes.
Right?
Yes.
So what do you think about that?
So I'm not sure the implication of the question is. So what?
While connecting, I just had a quick.
And each stem cell will have its own, if you like, lexicon, its own complement of mutations. And so the functioning of each stem cell will be slightly different, right? And as I said, if you get a driver mutation, then it expands. Now, the other point is, even leaving expansion to one side, how will those different stem cells work in their environment? So if you were in an environment that was, for example, a certain diet or a certain lifestyle, how would your blood stem cells, each one of those 20,000, that's all different, how do they interact with your lifestyle? I think that's the question that Stefan is asking. I mean, the problem here is it's going to take us generations to understand this.
Professeur Benoît Van den Eyde, pour votre question.
Il n'y a pas de question, hein, comme ça.
Pardon? Ah, vous n'avez pas de question? Ah, mais merci d'être venu, professeur.
Vous vous trompez.
On était ravis de vous entendre. Votre main était levée, désolé.
You have a question? Yes.
So what causes the difference between individuals, right? Why do some nations, individuals already have, clones at the age of 10 or 20, and others start much later?
So, I mean, the short answer is we don't know. But I think what is very clear is the date of acquisition of these clones could simply be a random stochastic process, right? That this is when the clone arose. I think more interestingly is, why do people's clones increase at a different rate? What's also very clear is that you could acquire a clone when you're 20 or 30, 2 or 3 different individuals, but at 70, the clones may be very different in size. Okay, and that's a really interesting question. What really determines clone expansion and clonal trajectory? I'm saying stochastic event. It's the time of acquisition of the mutation. Now, the other answer to your question, of course, we don't know, is what else in the germline, or what other somatic mutations helped acquire that mutation at a certain age 10 or age 30? We don't know that either.
And what about epigenetic changes? That's something that's difficult to measure, right? That could be there as well.
So what Ruud is saying is that in addition to genetic changes influencing either the acquisition of a mutation or the expansion of a clone, what about the entire epigenome? And so we think about cells having epialleles, just as we think about cells having genetic alleles. And as Ruud has mentioned, there are a large number of epigenetic changes. There are epigenetic changes in the histone code. There are epigenetic changes in terms of DNA methylation. There are epigenetic changes on the RNA. So RNA is methylated and modified. So measuring all of these different changes throughout the entire genome, throughout our entire transcriptome, is a Herculean task, and we don't have sensitivity to do it currently at a single cell level comprehensively. And so that's another Star Trek voyage.
Thank you very much, Professor. Acute myeloid leukemia is caused by combinations of genetic aberrations, among them the EVI1 protein. Using it as a molecular target, this is an option our second guest will develop in his lecture. Mr. President.
It's a great pleasure and an honor for me to introduce Professor Ruud Delwel. He has obtained his master at the University of Leiden and then a PhD in 1990 at the Erasmus University in Rotterdam on regulation of in vitro leukemia proliferation and differentiation. And then he moved as a postdoctoral fellow to St. Jude Children's Research Hospital, working with Dr. G. Miley, working on retroviral insertional mutagenesis in leukemia. And we discovered last night that we overlapped in Memphis at that time. He is now a professor of molecular leukemogenesis at the Erasmus Medical Center. He started there in 1993 as an assistant professor. He is very well known in the field for several major discoveries, such as that acute myeloid leukemia subtypes can be recognized by unique gene expression and DNA methylation signatures. Today, this is absolutely easy to think about, but at the time when this was found, it's a big advance, uncovering the abnormal expression of EVI1 as a marker for poor prognosis of AML, discovering how translocation of GATA2 enhancer regulates EVI1 oncogene, discovery of CBPA in AML and frequency in AML, and many other profound advances in the field of AML biology. And with that, we thank you very much for being here today.
Okay, thank you very much for the kind introduction, and thank you very much for the invitation to be here. It's an honor. The funny thing is, indeed, we just talked about yesterday that we overlapped at St. Jude's, and the interesting thing for me was, so I was a postdoc there 30 years ago. I just came yesterday from St. Jude's to give a lecture there, so that was really, was a big pleasure to do that. And then from there, I landed yesterday, took the train, and now I'm here. I haven't seen my wife yet. Anyway, so how good genes become evil in acute myeloid leukemia is the title of my talk, and I call it evil. It should in fact be a little bit. Maybe, right? This is the. Can you read this, right? So a very brief introduction. This is for those who are not very familiar with hematopoiesis. This is a picture of a normal bone marrow. This is a picture of a bone marrow of a patient with acute myeloid leukemia. You see clearly that there are major differences, right? And what you're looking at here is many cells in development, whereas here what is happening here is there's one cell. First, when we talked about those clones, is one cell that really, became leukemic and takes over the bone marrow sense, it's really the idea, right? The one cell that transforms. So this is the normal hematopoiesis. This one cell, has become leukemic and takes over the bone marrow and blood. When you look at patients with acute myeloid leukemia, patient A, B, and C, there's a difference. You can at the same time see, that they all have the same thing, we call it blast cells, accumulation of blast cells in the marrow. Through the microscope, they may seem very similar, but really live, it appears that there are differences. So some patients don't respond to any form of therapy, and they just die within a couple of months. Other patients can be cured, and there are quite a number of patients that seem to be cured, but then after a while, the disease comes back, frequently in even worse form. So AML is not one disease. There are many AML subtypes which can be recognized by recurrent cytogenetic or genetic mutations. Those recurrent defects are frequently predictive for therapy outcome, and I'm going to show you some examples of that. And so my talk will be much more molecular. And many of those breakpoints, so-called fusion genes, have been generated. Those fusion genes were first not present, but chromosome A comes to chromosome B, and at the breakpoint, there appears to be the gene of the one chromosome, the gene of the other chromosome, and you get a fusion gene, and that fusion gene appears to be transformed. This is different in the type of leukemia that I'm going to discuss with you today. So here I give an example. This is a picture from in a paper from our good friend David Brimwaite, who is not among us anymore. And what you can see here, this is based on cytogenetics. Approximately 40% of patients with acute myeloid leukemia can be recognized by cytogenetic defects. The other 60% have a normal karyotype, but have much more subtle mutations. The mutations that we have been talking about today, in fact, and heard about. And here you can see that based on those cytogenetic defects, there is clearly a difference in response to therapy. So what we try to do in our lab is really try to study the genetic and epigenetic defects, and also try to understand the biology of it. And we all do this all to better predict therapy response, to unravel the molecular biology of the disease, and we do this, and this is really, the purpose of our research, and that I would like to discuss with you today, is can we now kill those AML cells maybe in a much more specific manner, particularly there where current therapies are just not doing what they should do. And therefore, today I would like to focus only on one subtype, and that's this subtype. And I sort of, decided 20 years ago, after studying many patient subtypes, my research will be focused on one thing, try to understand this disease, and can we do anything about it? So patients with this abnormality are patients that have overexpression of a gene called EVI1. So that's why my title is, this is the evil gene, EVI1. EVI1 is located at chromosome 3q26, and it has two forms, an MDS1, a long form and a short form, and only the short form is important, that is this gene here. This gene encodes for a protein, it's so-called zinc finger protein. And zinc finger protein, there are zinc fingers here, these are pieces, amino acids, these are domains that bind to the DNA in a sequence-specific manner. They can bind DNA very specifically. There's another here, and there are some other regulatory domains. EVI1 is located at 3q26, and it is abnormally expressed in patients with either a translocation or an inversion involving 3q26 and 3q21. And I'm not going to show you all the data. I could have given you a talk about how we discovered this. I decided to do something else today, but the conclusion of many years of work that we did is the following. So patients have a normal chromosome 3, and they have this inversion, where so this part here is, there's a cut, it reverts and gets in again. Meaning that here, at 3q21, there's a gene called GATA2, which is a transcription factor very critical for normal hematopoietic cells. progenitors. Here is EVR1, and in the cells that are transformed, EVR1 is normally not expressed. It should be off. That's really the idea. However, as a result of this inversion, or also a translocation, a so-called enhancer, we have promoters and enhancers. Enhancers are elements in the DNA that can influence the activity of a promoter and thereby increasing the expression of the gene. GATA2 has, the break here causes that GATA2 donates its enhancer to EVR1, and on this allele, GATA2 is off, whereas EVR1 now is turned on, activated by this hijacked, we call this enhancer hijacking. And it appears to be a mechanism of transformation that occurs much more frequently than we previously thought it would happen. Okay, I come now to the questions that we try to address. So we have this gene, and we ask the question here, is EVR1 essential for growth and differentiation arrest of those leukemias? Can we show that? So we did a very simple experiment. This is a very old experiment. We have cell lines that we can culture, and there's a couple of cell lines that are transformed by EVR1. And there are, oh wait. And here you see two of them. It's called SB, and this, I mean, these are just names of cell lines. And this is a Western blot, and you see here that you can, by Western blot, show that these cells express the EVR1 protein. With shRNA, and nowadays you can also do that with genome editing, you can turn a gene off, very specifically. You can see that here by Western blot. This is a control cell line that has no EVR1. We did colony cultures, that is measuring the proliferative activity of those cells. And what you can see here, these are the two cell lines that proliferate. We switch off EVR1 and they totally lose the ability to proliferate. Meaning that these cells really depend on the presence of EVR1. If we do the same trick in a control cell line, which doesn't have EVR1, nothing happens. That experiment you do, because then you show that this is not an off-target effect, really knocking down EVR1, cells stop growing. This is a picture, flow cytometric analysis of a patient cell with an EVR1 abnormality. And what you can see here, this is CD34, is a marker at the membrane for very primitive cells. CD15 is a marker for cells that are very mature. And you can see that there are cells, they have all the 3Q abnormality. There are immature cells, a little bit more mature, and even cells that are CD34 negative, 15 positive. They all have the translocation. If we now take those cells and we knock EVR1 out, as I showed you already, we not only show a proliferative response, we also see that the cells now differentiate. So really the cells depend on EVR1. EVR1 causes that the cells are very immature, but if we can switch it off, then the cells start to differentiate again. When we sort those cells and put them in culture, only these cells were able to grow. These cells don't grow in culture anymore. So the question is, even though there is the translocation, is EVR1 expressed in every fraction or not? And we did so-called immunofluorescence by taking antibodies by which you can stain for EVR1 protein in the nucleus. And what you can see here, unsorted cells, in this fraction, EVR1 is present, but here, EVR1 is not present at all. So this tells us, first of all, that EVR1 is essential, right, for leukemia growth, but we can, if we can switch it off, the cells differentiate and they stop growing. And what we show here, that the cells really, when they differentiate, there is a way to switch off the gene, even though the translocation is still present in those cells. So we hypothesize that EVR1 may be a molecular target for AML with those abnormalities. And we think there are two ways that we can study that. We can either study by interfering with the function of EVR1 or by inhibiting EVR1 transcription. We have done both, but for the sake of time, I will only discuss with you interference with the function of EVR1. So what did we do? I'm really going to tell you now some, new molecular tricks, the most modern molecular tricks that you can do. What we did, we took our cells with this abnormality and we put there a so-called AID. Doesn't matter what it really is. The trick is the following. We now have a situation where there is a ubiquitination complex that we can bring to EVR1 with so-called auxin, and when you do that, within minutes, EVR1 can be eliminated, broken down. So we have this trick where we can very rapidly now degrade EVR1 in the cells that we study. And it goes really quick. Here you see a Western blot. We give auxin to the cells, and within 1 hour, there's no protein left. And this is a very pleasant way of doing the analysis, because usually we did shRNA, so you really have to wait 48, 72 hours. What you really would like to do, you want to degrade, get rid of EVR1, and immediately ask the question, what is the effect of eliminating this oncogene? So again, we first asked the question, and I showed you already, if we now delete EVR1 so quickly and we follow the cells and we keep doing that, what happens to the cells? And again, you see the cells very nicely differentiate. So there's really an effect. You degrade EVR1 and the cells start differentiating. And you see that also here in this analysis over time. So immature cells, intermediate cells, and mature cells. As I already told you, EVR1 is a protein that binds DNA. And we asked the question, where does it bind to the DNA? And what is the downstream effect? When you eliminate EVR1, what happens then with genes that may be either activated or inactivated? And to make a long story short, so we targeted EVR1 very rapidly, and then did so-called SLAM-seq, is a trick by which you can look immediately, rapid effect on transcription of genes. And we only found a handful of genes. There were 6 genes that were suddenly upregulated when you degrade EVR1. And among those 6 genes, there was one gene, for those who are familiar with what happens, which genes are critical for normal hematopoiesis, there's a gene called CBP alpha. And CBP alpha is a gene of which we know that it is frequently mutated in AML as well, in other subtypes. So we checked then, is CBP alpha indeed upregulated the moment you eliminate EVR1? So if you can see here, this is control, and here you see 3 hours, 6 hours, 24 hours, very rapidly, the CBP alpha gene is upregulated when you degrade EVR1. We also did it by Western blot. Again, here you see the Western blot, 3 hours, 6 hours, 24 hours, there's no EVR1 anymore, and in time, by Western blot, you see that CBP alpha protein goes up. CBP alpha is a gene absolutely essential to generate neutrophils. And the idea now is that EVR1 represses CBP alpha, and neutrophils cannot be generated anymore. Cells stay immature, and you get leukemia blood cells. Then the question is CBP alpha really such an important downstream target of EVR1? Then we should be able to bypass the effect of EVR1 by introducing CBP alpha. And that's what we did here, so-called DOX-inducible system. Again, another molecular trick to rapidly now not degrade, but activate the gene. Here you see the cells, the leukemia cells I showed you already. And when we degrade EVR1, right, this is happening. Now we don't degrade EVR1, no, we overexpress CBP alpha. And again, you see the same thing happening, the cells differentiate again. And this is shown in a different way. Does EVR1 repress CBP alpha expression at DNA and at chromatin? And I have to tell you one thing now. So now we look at the locus. So what does EVR1 really do? I told you EVR1 binds the DNA. This is a so-called ChIP-seq experiment. Now we really look at the genome. This is the CBP alpha gene, and here is an enhancer of CBP alpha, which is 40,000 base pairs away from the gene. And what you see here is that EVR1 binds here. So what is this region where EVR1 binds? So we have a look now, what is the function of that enhancer? I think I'm going to skip this for now. What I want to show you here is this is the CBP alpha gene, and CBP alpha gene is normally regulated by this enhancer. This enhancer is bound by many factors, transcription factors, that activate this enhancer, forms a loop with CBP alpha, and then CBP alpha is expressed. And it's exactly at this important region where EVI1 is bound. If we mutate this enhancer, where EVI1 is bound, if we mutate that, you see that CBP alpha expression is gone. And we look at the neutrophils now. So here you have wild type mice, and here you have mice where the enhancer is mutated. This again is staining, flow cytometric staining of neutrophils. This is in peripheral blood and in bone marrow. You have nice neutrophils. And when you delete this enhancer, all the neutrophils are gone. This is the place where EVI1 is binding. So the question is, if you degrade EVI1, what happens at the enhancer? And I go quick here. When you degrade EVI1, the enhancer becomes active again. This is HTK27 acetylation. There's a mark for an enhancer that becomes active. It is bound by a so-called P300 protein, and also transcription factors bind with higher affinity and more frequently at this enhancer. So what we show here is that this particular enhancer is bound by EVI1. When you degrade it, CBP alpha goes up and the cells start to differentiate again. Well, keep in mind that this gene is so bad that the leukemia cells don't respond to therapy. So the idea is, can we now use this information to target EVI1 and to activate transcription again and differentiation again? So how does EVI1 repress when it is bound to the enhancer? How does it do that? So here we did another set of experiments. We took the cell, leukemia cells, and now said we pull down EVI1 by an immunoprecipitation and do mass spectrometry and ask the question, which other proteins bind to EVI1? And we found that EVI1 binds to a protein called CTBP. I'm going quick here. What I really want to show you here, we made all kinds of mutants of EVI1 and found that in EVI1 there is one tiny piece of protein here, just 5 amino acids, that are able to bind to this protein CTBP. And when you make a mutation of that, you lose this interaction. This piece of protein, this peptide, is called PLDLS, is very important, and it binds this CTBP. And when we introduce EVI1 in mouse bone marrow cells, what you see here is they form colonies, right, in yellow. And they keep growing, much better than cells that have no EVI1. If we give the mutant, then we see that this proliferation is diminished. So we decided, and here comes really the trick of how can we now treat leukemia cells with this abnormality. Can we now find ways to interfere between the interaction we see between CTBP and EVI1? So we made peptides, PLDLS. We know that that binds, this piece of protein binds to EVI1. So we give peptides, PLDLS peptides. And by that, you compete between EVI1 and CTBP. This interaction is not present anymore, and we predict that if we do that, the leukemia growth will be interfered. Will not happen anymore. As a control, we use a peptide with a mutation in it, which should not be able to do this. So we took bone marrow cells that were transformed with EVI1. They form colonies, and they keep growing. When we give this PLDLS overexpression, you see that we completely interfere with the proliferation of those cells. whereas the control has no effect. And when we look at differentiation again, shown here, this PLD, so these are the cells transformed by EVR1. When we give this PLDLS inhibitor, we call it, the cells nicely differentiate again, and also when you look at bone marrow smears, you see neutrophils back. Whereas here we have blast cells, still leukemia cells. Here you see very nicely that if we look in the leukemia cell, human leukemia cells, you see this really tells you that there is interaction between EVR1 and CTBP. When we give this PLDLS, you totally lose the interaction between the two proteins. And that is also shown here. So to make a long story short, there's EVR1 that binds through this domain a so-called CTBP, and CTBP recruits many other proteins that really affect the chromatin, and we call this co-repressors. And when you give PLDLS, CTBP is not bound anymore, the co-repressors are gone, and EVR1 CTBP interaction is not happening at the DNA anymore. So we took the human cells now, and again show that if we give this PLDLS, you see that CBP alpha goes up, you see that colonies are inhibited, and we did even a transplantation experiment, shown here. So we took mice, we gave the leukemia cells either control or with the PLDLS inhibitor, and you can see here very nicely, the leukemia growth is quite heavily here in those mice. In the controls, but when you give this PLDLS peptide, you can see very nicely that this growth is inhibited. So what we think is going on here is that so the cells are transformed by EVR1, overexpression. PLDLS is an important interactor for CTBP. There are co-repressors, and if we are able to interfere with those two, then the cells would differentiate again into the same situation as if EVR1 would not be there. I don't think I have, this is the end of my talk. One thing that I have to tell you, we have now developed 2 different assays by which we are going to try to screen for compounds that may interfere between EVR1 and CTBP interaction. That can be an alpha assay, a biochemical assay, and we have also a sort of in vivo cellular assay to really identify small molecules that are able to interfere between the 2 proteins. And hopefully, find something that is at present, an incurable disease. With that, I would like to thank all my colleagues from Rotterdam and from other institutes that are very important in this study.
Thank you very much for this very interesting lecture, and I cannot stop myself asking, how does that peptide enter the cells?
Okay, and I haven't explained well. This is a construct. It's a construct. So we did a 4-time repeat construct. So it's a construct that we introduced into the cells now.
Mini gene, or?
It's a mini gene. So one thing that should be possible, because we have done that in the past, is you can make small peptides, for instance, with a TET moiety on it, so that you can get it into the cells. But I mean, these experiments take 6-7 days, and that is then the difficulty there really to study differentiation.
Questions from the room? Thank you. Questions? Pierre Coulis? Yeah.
What are the expected issues if we block CTBP binding to other targets, possibly?
To other proteins. I mean, I understand your question, and I think that question can be asked probably for every type of therapy that we do, right? You try to find a window which will be useful to apply this. I understand that what you're saying, although there are not very many other proteins that bind CTBP. Another issue maybe, I didn't explain that yet is that EVR1 itself, of course, has also a normal function, right? And it is present in dormant hematopoietic stem cells. So the dormant hematopoietic stem cells have EVR1. When stem cells start to proliferate, EVR1 is turned off. And that's why in the leukemia cells, it's normally not present. So the leukemia cell is just a little more beyond the dormant hematopoietic stem cells. So if you would be able to find molecules to target, you know, there is the possibility that you also target hematopoietic stem cells, but which are not proliferating, and maybe because of that reason, you may be lucky and not have a major effect there. But it is something that we have to test, yeah.
Other questions? Yes.
Do you know why this leukemia has micromegakaryocyte? This leukemia has micromegakaryocyte exclusively. Do you know why?
Why it has megakaryocytes? Probably because the cell that is transformed is primitive enough to go into the different directions. It is maybe a bit like BCR-ABL, right? BCR-ABL is also almost a stem cell transforming gene, and you get erythroid megakaryocytic. You find the abnormality at different lineages, and I think this is the case here as well. Yeah.
Maybe just to add to that, there's a lot of evidence now that blood stem cells are very heterogeneous in terms of the blood cells they produce. So not all stem cells will produce all blood cells, right? So that's a kind of paradigm shift. We always thought a stem cell produces everything, but no, certain stem cells are much more likely to give certain blood lineages. And the cells that are closest to stem cells are actually megakaryocytes and platelets. There's a direct line from going straight from a stem cell into a platelet. A lot of evidence for that. So I suspect the EVI1 transformed stem cells probably may have a megakaryocyte bias.
In fact, it's one of the few leukemias where, so in most AMLs you will find, you know, there is a major effect also on platelet formation, for instance, but they have normal platelet levels. And it's one of the few AMLs with normal platelet levels. Yeah.
There are no more questions from the room. Let's go to the Zoom. No? No?
So just one question for Ruud. So I think it's a fantastic example of how you really need to understand the biology of a key transforming mutation in any cancer. And did you ever systemically express your PDLS construct in mice? Just to think about the tox question, the toxicity question. So you've used a construct to put it into your MUS3 cell line, but I wonder if you ever widely expressed it in mice to see, is there a tox profile? Because it would help answer that question.
It would be a good experiment to do. No, we haven't done that, of course. No. But that may be an interesting approach to find out, you know, how major the effect would be if you do this in hematopoietic stem cells, right? Really. Right. Because I think you should then really do I would then, I think, force the expression into the hematopoietic system first.
Yeah, you could do either a expression within blood cells, blood stem cells, using an SCL cassette, for example, or widely in the mice. Yeah. And I think that may tell you something about how, when you develop your small molecules, how to develop them.
Yeah.
Now, in the biochemistry textbooks, we learn that when you flip enhancers, it's fine. So promoters you cannot reverse, but enhancers you can. But in your system, you probably inverse too much. It's not only that the sense of the enhancer is changed, but you completely brought the enhancer closer to the promoter, right?
Well, and you mean the translocation itself? So we identified many other translocations, by the way, now, right? So it's translocation 3A, 317, and every time an enhancer comes from that locus to EVR1. What is important, you know, that this is really detailed, is the CTCF sites. Because some of these enhancers are still 1 megabase away, but they are within a topologically associated domain.
Eliminate the frontier.
Yeah, but it is the CTCF site that really brings the enhancer in the vicinity to the promoter. And the orientation of the CTCF site is very important.
Okay.
If that is not correct, then nothing happens. And that's why the orientation of the enhancer together with the CTF does matter.
I have no question at the moment, but I will remind you, if you are on Zoom, that you can still raise your hand or transmit some questions through the chat. Thank you.
Good. So if there are no more questions.
Okay.
Thank you very much.
Studying the structures of the brain-derived fibrils to better understand the pathways to their formation and provide new targets for the development of therapeutics for Alzheimer's disease, as well as for cerebral amyloid angiopathy. This is the research the next speaker is going to present. Mr. President.
Thank you. So it's a great pleasure for me today to introduce Professor Stephen Smith. We collaborate for probably almost 25 years. He's going to speak today about a different field than the one we collaborate, but he is an expert in protein folding and especially transmembrane protein biochemistry and structure. He has obtained his bachelor in chemistry in California State University, Humboldt, in California, 1979, and then he went and obtained a PhD in chemistry in 1985 at the University of California, Berkeley. He then worked at the Department of Chemistry at the Woods Hole Oceanographic Institute in Woods Hole, Massachusetts, then back to University of California. His advisor was Richard Mattis, and he worked on well, things I cannot even pronounce, resonance Raman studies on the mechanism of proton translocation by bacteriorhodopsin retinal chromophore. He then went and did a postdoctoral fellowship with Professor Robert Griffin at the MIT, where he also worked with Professor Gobin Khorana, who at the time was interested in rhodopsin. He is the person that solved the genetic code. And then he became assistant professor at Yale University, then associate professor at Yale, and then went to Stony Brook as a professor and chairing the structural biology department. He's a director of structural biology since 1998. Well, thank you very much for being here, and we look forward to your talk.
So as with the other speakers, I'd like to thank the Academy and to thank Stefan for the invitation to come. Can everybody hear this well enough? So I'm going to switch gears a little bit from talking about molecular sides of biology, switch gears from cancer to talk about another important disease, namely Alzheimer's disease. But sticking with the topic of hematology, I'm going to mention, I'll talk a bit about the vascular component of Alzheimer's disease. So as part of the background to my talk, I'm going to go through what the major players are and talk about the work we did in collaboration with the groups here in Brussels, and then talk about how that translates or has carried on to our more recent work. So there are many players in the progression of Alzheimer's disease that are the two principal players that form fibrils in the brain are, where is the pointer? Are the tau protein, and the tau protein is involved in microtubule formation, and what happens is that with this protein, hyperphosphorylation leads to the polymerization into fibrils of this protein to form neurofibrillary tangles within the cell, which leads to cell degradation and eventually cell death. The other player, which is the one I'm going to talk about, is comes from the amyloid precursor protein. This protein gets degraded by two proteases, beta secretase and a gamma secretase, to release A beta peptides. These are short peptides that are normally cleared or degraded in the brain, but in Alzheimer's disease, these peptides associate into fibril forms in the plaques. The process of fibrillization goes through a series of intermediates involving first associating into small oligomers and then into protofibrils. And this is important is because these oligomers are thought to be the major player in memory loss, binding to receptors in the synapses of cells here, or forming pores or otherwise degrading the membrane, which leads to cell death, neurodegeneration. The two major peptides, the A-beta peptides come in different lengths. The major peptide is a 40 amino acid peptide, A-beta 40, and then the other minor product is the A-beta 42 peptide. It's the A-beta 42 peptide that's the principal component of these parenchymal plaques in the brain. However, and it's the case that an increase in the A-beta 42 to 40 ratio is associated with AD progression. However, there's a second disease called cerebral amyloid angiopathy, and this is where the A-beta 40 peptide principally forms vascular amyloid on blood vessels, cerebral blood vessels, which leads to the degeneration of the blood vessels, hemorrhaging, can lead to stroke. There's a big component of inflammation with these amyloid plaques that are being formed. These two diseases often occur together. Most patients with Alzheimer's disease, about 80% of patients also have cerebral amyloid angiopathy, CAA, but it is also distinct, so it can occur in the absence of these vascular plaques. As with Alzheimer's disease, there are familial mutations that lead to the enhanced deposition of this vascular amyloid and early onset of this disease. So what we've been interested in is, from a structural standpoint, what leads to this increase in the 42-40 ratio and AD progression, what leads to, by these mutations in cerebral amyloid angiopathy into the vascular deposition, why the difference between 40 and 42, and then also what's the role of oligomers and fibrils in terms of the toxicity to neurons. And so what we've done is we've taken a structural approach, and we've looked at everything from the amyloid precursor protein, the protein that generates the A-betas. We've looked at the monomers, the oligomers, the protofibrils, and the fibrils. I'm going to first talk about our early work, mainly in collaboration with the group in Brussels, looking at the amyloid precursor protein, and then I'll transition to talking about fibrils towards the end of my talk. So the first part has to do with looking at the amyloid precursor protein with Stefan's group, the group of Jean-Noël Octave and Pascal Klein-Lin-Campard. So the amyloid precursor protein is sort of diagrammed here, has a single helical transmembrane domain. It has a large extracellular domain, and the A-beta peptides are created by first cleavage to get rid of the extracellular domain, and then an intracellular cleavage to clip off the intracellular part. It's this part then that forms the fibrils. This is the sequence that shows the transmembrane domain, the original cut site to form, to begin to cleave. So the first cut site is up here called the beta cut site. The A-beta 40 peptide are these 40 residues here. To generate this peptide, there's first an enzymatic cleavage here, and then it progressively gets cut to shorter and shorter sequences, and eventually this is all hydrophobic. It's no longer sufficient to span a cell membrane, comes out of the cell and begins to fibrilize. What I want to point out, and it becomes important, is that the A-beta peptide, the N-terminus of this is mostly hydrophilic, and the C-terminus is hydrophobic. Yet there's this one stretch of hydrophobic residues that's really important, sort of in the middle of the protein, sort of middle to the front of the protein. It has this sequence LVFFA. That'll become important throughout the talk. So the question is, what are the structural features that may determine these cleavage events? And what we determined, in this case using NMR spectroscopy, looking first at the transmembrane domain, is that the transmembrane part of this sequence forms an alpha helix. And these alpha helices actually dimerize through a glycine, XXX glycine motif. At the gamma cut site, this is helical. It's actually not accessible to proteases. Whereas structurally, you can show that where the epsilon cut site is, this is where the first cut is. The enzyme active site is at the membrane surface. So structurally, looking at this sequence, that makes sense. Between those cut sites, in this region, as you progressively cut up to this region, this is where a number of familial mutations occur that control exactly what the length of the peptide is. So it's these are a set of mutations that will end up changing the dimerization rate, but also change how the substrate interacts with the enzyme. So the next thing we do, that's the transmembrane domain. The next thing we did was to try to look at the soluble extracellular part, the hydrophilic part of this protein. And that part, beta, forms both alpha helical structure with this big peak here. I'm not going to explain this data. And beta sheet structure. But there are mutations. So what Stefan's group did was they sequentially mutated the extracellular domain with alanine. And what happens is that there are places where if you mutate, in particular this LVFF hydrophobic sequence to alanines, you remove that beta component. And the result of that is that the processing of this peptide to form the A-beta peptide shoots up dramatically. So there's structurally what we see is that you have transmembrane dimer, and then as you come out of the membrane, the N-terminal part of this forms beta sheet. So this beta sheet lays on the surface of the membrane, and it's this hydrophobic LVFF sequence that tethers the sequence to the membrane. Between the transmembrane domain and this beta region are these residues GLU22 and D23 that are hotspots for familial mutations that lead to this disease, cerebral amyloid angiopathy. Beta sheet, hotspot for disease mutations, alpha helices. So structurally, this is what we knew about the amyloid precursor protein. If you remove this beta sheet and have this dangle out, then this is more accessible to the enzyme, and you get an increase in the A-beta peptides that are formed. So before I talk about the fibrils, this goes back to the original data that got me interested in this field. As in the introduction, the bulk of my research is on G protein coupled receptors. Has nothing to do with Alzheimer's disease. And it was my wife, whose family has a history of Alzheimer's disease, that said I should work on something more important than GPCRs. And so I started, structurally we were looking at membrane proteins, and the protein that we're using as a model system for developing structural methods for looking at membrane proteins is this red blood cell protein glycophorin A, which spans the membrane with a single alpha helix, and we could show dimerizes through these glycine XXX glycine motifs. So as a place to get started, what is really striking about the amyloid precursor protein, or APP, is that it has 3 of these motifs in a row linked together. And so does the human prion protein. And in fact, they all have a GXMXG motif. So thought that was a good place to start. And what this glycine motif does is, when this is in transmembrane helices, the glycine is perfectly tolerated in helices and forms notches that the helices can associate with. When these helices transition to beta sheets, so the fibrils in the A-beta peptides are made of beta sheets, so you have an up-down structure. In this case, the glycine's on the same face of the helix. For the beta sheet, the glycine, methionine glycines are actually on the same surface of the beta sheet. And these things polymerize in a parallel and register orientation. So you make the fibrils by monomers associating. And what that does is it leads to these grooves on the surface of the fibril. And so the first thing that we did was, since glycophorin A had these motifs, we asked the question, does that fibrilize? So we actually cut this protein in the same place where the APP would be cut, and lo and behold, nicely forms fibrils. And what we first did is then determine the structure of those fibrils, and what happens, and why these things form very, very stable fibrils, is that the methionine packs into the grooves created by the glycines. So the very stable fibrils are formed like a Ziploc bag that these ridges fold into these grooves. And after realizing that, what began to come out from a number of different groups were structures of fibrils. And here is A-beta 40. A lot of this work has been done by Rob Tico's group at the NIH. So these are, the fibril axis is in the middle of the page here. This is a monomer. It forms this U-shaped structure. The methionine glycine grooves are at the interface between these two proteins. But the thing that I want to point out is that how these things fold is that you have the hydrophobic C-terminus and this hydrophobic LVFFA sequence glumming together. So when you add A-beta peptides to solution, the two hydrophobic parts come together to form these U-shaped structures, and then that forms the nucleus for polymerization. That happens for A-beta 40. This is early work on A-beta 42. A-beta 42 actually has a different structure than this. The C-terminus folds back on itself to have 3 beta strands. But the general idea of having these U-shaped structures with the two hydrophobics coming together holds. So having found that out, we and many other people went through lots of efforts to look at inhibitors. First working with peptide inhibitors that would fit into these grooves and prevent the fibrils from coming together. Easy to do. And upon learning or seeing this, what we found is that there's a small molecule curcumin, which is the yellow pigment in turmeric, that actually has 2 aromatic residues that has exactly this spacing. So if you add curcumin to fibrils in solution, they'll dissolve. I solved the problem with my wife. I told her all she needs to do is make more chicken curry with lots of curcumin, and I could go back and start working on G protein coupled receptors. I'm being a little bit facetious, but what we then did, the problem is a little more complicated. We began to look at the fibrils in a much more detailed fashion, and this brings up the second set of collaborations that are ongoing today with two groups. Bill Van Ostren's group, Bill works on, used to be at Stony Brook, now he's at University of Rhode Island. He works on transgenic mouse models of Alzheimer's disease, and Annemieke Roosmuller, who's in Amsterdam, that works on human patients. She has access to tissues at the brain bank at the medical center in Amsterdam. And so what we've been trying to do is not look at fibrils in solution, but to look at actually what the fibril structures are in human brain, both in sporadic CAA, sporadic AD cases, and in disease form cases. And the way that we've been doing this is to use a technique called laser capture microdissection. We get human brain tissue, identify where the amyloid is by using a fluorescent marker, and then use laser dissection to specifically cut out that amyloid. Once we have that, we can look at these are TEM images of the fibrils in sporadic CAA cases and in a Dutch patient. And you can see these are dense fibrillar networks. These tend to be short fibrils. These are longer fibrils. But what we can do with this tissue is we can then propagate it. These are just biophysical techniques of infrared spectroscopy and fluorescence spectroscopy, showing that you can take and use the brain fibrils as seeds to grow more fibrils for structural studies. So trying to determine what the fibril structures are. And just by looking at TEM, one of the things that became clear is when we seeded off these brain samples, we got some really unique looking fibrils. These ones that are extremely twisted were something that came out of these samples that we had never seen in vitro samples. And so we've been using cryo-electron microscopy to determine their structures. This is the structure, say, of a twisted fibril, has a certain width, and then a certain crossover distance to how twisted it is. And we can characterize morphology by looking at the crossover distance and width. And when we do that, this is just now comparing two fibril forms, a sporadic CAA case and a case that had the Dutch mutation, which is E22, G. And what we find is that the samples are pretty heterogeneous, but in both cases, we find this unique, highly twisted structure that the population is much more presented in the sporadic case and much less so in the Dutch case. And then there's a second population that's pretty well defined in the Dutch case with a defined width, but has variable crossover distances. So with cryo-EM, looking at those structures, one of the things that became immediately clear was what was unusual is that the N-terminus of this, which is in vitro solution fibrils, is usually unstructured. The fibrillization happens through the hydrophobic C-terminus. In this case, the N-terminal part is structured. Here's the C-terminal part. So this is, rather than forming a loop like this, here's one, the fibril axis is going down the middle of the page here. N-terminus is structured. Here's the C-terminus. Here's the sites that mutation leads to CAA. Here's one monomer, here's another monomer. This fold actually was determined by another group before we did this, using a different approach for extracting fibrils from vascular amyloid. Our structure is fairly high resolution. I just call it almost atomic resolution. The other structure was at much lower resolution, but the basic fold was the same. The interesting part here is that rather than having the LVFF sequence, this hydrophobic sequence, within a monomer interact with each other, this interacts with the C-terminus of another peptide. Okay, and so the interface between these two protofibrils is driven by hydrophobics. The folding of this one leaflet of the thing is driven by electrostatics, both between the loop here and driven by these residues, the E22, D23, the N-terminal aspartic acid in this lysine 28. So there's this charged cluster that keeps the N-terminus tethered. Okay, and that's just a closer view of showing these molecular interactions in those charge groups. So when you look at those charge groups, when the fibrils form, this is the view before, this is turned 90 degrees, the interaction between this lysine and the aspartic, which holds down the N-terminus, is staggered. And this is sort of important because the rapid polymerization of these fibrils relies on this staggered structure where you can have a monomer sort of insert and interact, electrostatically with the next residue. That was population A. The second population that we also determined the structure to high resolution, population B, is shown here. This is the electron density for that population. That actually had a structure similar to a structure that was not derived from vascular amyloid, but from parenchymal amyloid previously. So that you had the peptides with the two leaflets here stretching out, but also density on both sides of those that couldn't be defined within the structure and was assigned to beta hairpin, sort of a monomer sticking to the outside of the protein. The important point here is that, that like the previous structure, this structure also has the LVFF sequence sticking not within a monomer to the C-terminus, but across the interface with the C-terminus. Okay? So what's happening in this case, so this is just showing the LVFF sequence here, interacting with part of the C-terminus, and then the idea in this previous structure was that the beta hairpin, the hydrophobic portions of the beta hairpin would stick to that to hide those hydrophobics. So the electrostatic residues, you have this similar cluster of residues, but it's no longer holding down the N-terminus. And it's no longer staggered. The idea behind this is that in population A, the N-terminus is stabilized by these electrostatic interactions, and in these familial mutants, this interaction is broken, and the N-terminus can flop open. Okay, so one of the conclusions, and this is on my last slide, but I'll get to it here. One of the conclusions from this work is that the fibril structures that we see in the brain are different from those that you would get in solution. The reason that this makes sense is, for us, is that, as I showed you previously with the work on the amyloid precursor protein, the fact that these things have a beta sheet on the N-terminus due to interaction with the membrane and the C-terminus coming down into the membrane means that when you do the final cut and it comes out, the N-terminus is already folded. So the LVFF sequence is not available to stick within a monomer to that sequence, right? Rather, it's starting the folding process for making the fibrils depends on the membrane that's there. Why that's important is that there's a mountain of work of people looking at structures of oligomers, fibrils, protofibrils, where they take A beta peptides, synthetic, add them to solution, and use those to develop inhibitors, antibodies, pharmaceutical agents. And I think it's going to be this realization that the pathway of formation is different in a cellular environment that's going to be important. We've been mutating those residues and then looking at fibril formation, and without going through these slides, that general idea that they're important for guiding the fibrillization in the brain holds. And to finish up, my point is that the solution mechanisms are different than the cellular mechanisms. I would argue that the N-terminus is going to be a better target for pharmaceuticals than the C-terminus. And in fact, the antibodies that have been produced to date that have gone to clinical trials target the N-terminus, and it usually targets an unstructured N-terminus. Knowing what the structures are will allow one to perhaps get more selective small molecule drugs and perhaps antibodies to target these molecules. And then finally, going back to the idea of fibrils and oligomers as to what's the toxic species, it's probably difference between Alzheimer's disease leading to cellular toxicity of neurons and CAA, where the A beta peptides lead to degeneration of the smooth muscle cells surrounding blood vessels. And I'm just going to leave with, I just love these images that show, these are smooth muscle cells from the cerebral vascular of human patients in culture that show fibrils forming and penetrating the cell surface. This is A beta 42, this is the Dutch peptides, so the fibrils will form, they start to intercalate into the cells, and if you follow this with time, the cells degrade. So this has to do with the process of fibril formation and not oligomers forming pores. So trying to understand the differences of these two diseases, trying to understand how they develop structurally is an important component to perhaps looking for better therapies. And with that, there's a number of people that have been involved in this work at Stony Brook, here in Brussels, Rhode Island, and in Amsterdam. Thank you for your attention.
Thank you very much for this very, very interesting talk. Jean-Pierre, yes. Jean-Pierre Brion.
Hey, thank you, Professor Smith, for this beautiful talk. It was a beautiful study, biophysical study on the fibrils. I wondered, in your opinion, what's exactly a protofibril? Because, I mean, it might be very important because, as you know, the last antibody, the lecanemab, is supposed to target protofibrils. And I to have higher affinity for the protofibrils compared to the monomers or the very aggregated fibrils. But this has therapeutic implication. What is a protofibrils in your opinion?
I can have the slides back. So I have control of it. So when you add A beta to solution, what this slide shows is these little round dots here are the oligomers. These squiggly things that are usually about 100 nanometers long, those are classically protofibrils. And then these will transition into fibrils. So how I would describe protofibrils are these squiggly things that result from association of oligomers. And we've studied that transition from oligomers to protofibrils. The fibrils themselves are typically formed by these layers of polymerized filaments, and those would be called, in terminology, protofilaments, to be distinguished, so that you might have 2 or 3 protofilaments associating and twisting to form a fibril. But the protofibrils are these aggregated, these linear, twisty, aggregated oligomers.
Pascal.
We could go into, I could talk for hours on that.
So Pascal Kinlan-Campar.
Thank you, Steve. In much of the work that you have been showing here, sometimes there are mutations that are used in order to induce these fibrils or study the effect of mutation, and often they are central motif mutation of the A beta domain. So we know that they are changing the folding, that they are changing the interaction or possible interaction that you have in the fibril formation. But the question is, regarding the sporadic cases, do you think that the changes in the brain context or cellular context that we might have are driving a mechanism toward what you are looking with the mutations or not, or what could be the critical aspects of the brain context that is indeed driving fibril formation? You have been talking about lipid context, you have been talking about membrane. So what would be to you the main point to investigate in order to have a good idea about what are the additional mechanisms from the context that we could explore in order to understand better the fibril formation without any kind of mutations?
I'll give you one example. So again, it's a complicated, there are many components here, but one example would be there's a role for metal ions in the progression of AD, and perhaps it's leading to different effects. The metal ions typically bind to the N-terminus. So if, in these cases, in a membrane environment where the N-terminus has folded into a beta sheet, where the electrostatics, the histidines, and the residues that would coordinate the metal ions are not accessible, you won't get metal ion binding. When the N-terminus folds out, as in this population B, and is unstructured, now that's a site for metal ion binding and shifting the populations between A and B, with the idea being that once you've gotten rid of the N-terminus, now the hydrophobic C-terminus has a much better propensity to aggregate and form fibrils. So it may be the case that an important component and a link in this whole story are copper, for instance, 2+, coordinating with the N-terminus and leading to enhanced fibril formation. But there could be, I could talk about other examples.
If I remember correctly, the mouse sequence doesn't induce the initial steps. There's something in the N-terminus of the mouse sequence, right, that changes.
And the question, yes, so we've looked at that a lot, and I can't give you a direct answer that would map one-to-one with these studies. And it's not entirely clear whether those mutations really affect the fold and the polymerization of the fibrils, or just influence the expression of the fibrils, or the expression of the A beta originally. And I think it's the latter. And so there actually isn't a structural role for those mutations.
Right, Paresh.
Steve, just a very sort of naive question. So imagine you were able to find inhibitors of fibrillization, end-amyloid inhibitors as you were describing. Would they dissolve preformed fibrils, or do they just prevent the formation of new fibrils?
One of my favorite examples is this idea of curcumin, right? This molecule that in solution they will dissolve fibrils. These are formed fibrils. The problem with curcumin is it doesn't cross the blood-brain barrier very well, which is why you have to eat lots of curry. But having said that, there's a group that engineered in a mouse model molecule that was able to transport curcumin into mouse brains that were loaded with plaques. And it did, in fact, dissolve those plaques and lead to better outcomes in terms of maze and cognitive studies. So if you can get it in, it has an effect. The issue has always been the toxic component has thought to be the oligomers. So if you're taking fibrils and converting those into oligomers, is that a good thing or a bad thing? With these antibody studies, the idea is that if you're binding antibodies to the fibrils, these things now have a bigger epitope to be recognized by microglia and begin to be just degraded internally by the brain immune system.
Jean-Noël Octave.
Thank you, Steve, for this very interesting presentation. I was wondering whether lecanemab, which the recent antibody which looks very promising in recent clinical trials, is also recognizing the N-terminus. Is that known?
Sorry, which is recognizing?
Lecanemab.
Yes.
Is also recognizing the N-terminus?
I don't know.
Okay.
So I assume that it is, but specifically I can't tell you.
Okay, thank you.
So this is the Biogen antibody that was made because a person made antibodies to it, right? Or it was, yeah. Okay, turning to the digital world. Well, if not, I will use the 1 second now to give to the 3 speakers the present of the Academy, which is a book about the Academy. The Academy has a great history. It's not only characterized by technical failure of the digital equipment, but also very nice architecture. If there are no, yes, questions? No. Right, questions? No. Yes, Inami. So people who have been on the Zoom.
So, si vous nous suivez sur Zoom, si vous nous avez suivis sur Zoom ou sur YouTube, que vous êtes médecin ou membre de l'Académie, eh bien, c'est le moment d'envoyer votre petite demande Inami. Alors, on n'a plus d'écran, donc je vais vous rappeler l'adresse que vous connaissez déjà tous par cœur, mais je vous la rappelle quand même. contact@armb.be, donc je répète, contact@armb.be. N'oubliez pas de préciser votre nom, votre prénom et bien entendu votre numéro Inami. Avec cela, eh bien, je vous remercie de nous avoir suivis au courant de toute cette belle année. On vous donne rendez-vous en 2023, précisément le 29 janvier, pour l'accueil des nouveaux membres belges et étrangers. D'ici là, passez de très belles fêtes. Prenez soin de vous. This is the end only on YouTube for the broadcast. Thank you so much for your time this morning. Have a nice holiday session. Ah, but no, actually, we don't have the communications. We already did the communication. So no, we are not going to stop the streaming. I will leave the floor to the president.
So thank you very much. So on the 28th of January, we will have a session where newly elected members, both Belgian and foreign elected members, will be presented. There will be a film about our new president, Professor André Shannon, and there will be, in the presence of the rectors of the Belgian Walloon universities. This year we will have the presence of the rectors, of the General Secretary of the FNRS. So I'm looking forward for an absolutely top digital electronic thing, because there's going to be a lot of authorities and members of the government. Several ambassadors will be here. So this was a training session, but next time it will be perfect. And now it's my pleasure to give the last word to our permanent secretary.
Pour vous souhaiter évidemment une excellente fin d'année, de bonnes fêtes pour vous et votre famille, et vous inviter au verre de l'amitié que nous allons avoir aussi pour fêter notre président, dont ce fut une très belle année et une magnifique session ce jour avec des orateurs prestigieux. Merci beaucoup.
Et sur Zoom, on est désolé de ne pas pouvoir vous offrir un verre et de le partager avec vous pour nous faire pardonner et vous remercier pour votre patience. Au revoir à tous.